So to begin with, Plasmodium, commonly known as malaria, is a genus of single-celled parasites. It's carried by mosquitoes and infects humans and other animals, and it's an extremely infectious and dangerous disease. It causes all kinds of terrible side effects and in many cases death. In 2010, there were over 200 million cases worldwide and an estimated 700,000 deaths, and it's particularly widespread in the developing world. So there are currently many strategies employed to deter the spread of malaria and to treat infected patients, and these include the use of bed nets and sprays and repellents such as DDT. There are some drugs such as chloroquine, and there's also some kind of further out ideas such as gene drives that John's going to go over later in the talk. But the state of the art right now is artemisinin-based combination therapy. So artemisinin is a highly efficacious drug derived from the plant Artemisia annua, or sweet wormwood. And it's kind of interesting because this plant was originally discovered through the use in traditional Chinese medicine, but it's been since proved to be a potent modern medicine as well. So there are a few major problems with artemisinin supply that lead to instability in providing enough treatments worldwide. It's difficult to grow, can only be grown in specific places, and actually takes 2 years to cultivate. So these have led to shortages in the past, which is a really big problem. There's also a limited number of suppliers, which has turned the medicine into a commodity with large price fluctuations and suppliers hoarding material and waiting for prices to increase. So as I said before, it's a very big problem for the developing world. So because of these problems, people were looking for another way to supply artemisinin for malaria treatments. So to quickly recap some of what Dan just went over, let's recall that life depends on DNA to code for proteins. Molecular biology techniques have allowed us to manipulate DNA in useful ways, such as expressing useful proteins like insulin. One thing we didn't go much into was what proteins do within the cell. So one of their important functions is to sustain the metabolism of cells. So metabolism is basically the chemistry of life. So proteins help make the chemicals that cells need to live and grow, such as caffeine, ATP, and ethanol. And proteins that carry out chemical reactions are also called enzymes. So metabolisms in cells are incredibly diverse. This is just a tiny snippet of some of the proteins involved in one specific pathway in a cell, and we are constantly researching more about how organisms break down and build up different chemicals as part of their metabolism. And we have much more to learn. If it were easy to understand, we'd probably have a much better idea on how to get super skinny or super buff really quickly, but we're not quite there yet. One day. So with the advancements of molecular biology techniques, people began to wonder, what if we could engineer the metabolisms of organisms to make useful chemicals? So the basic premise is not really a new one, and some of you have probably benefited or suffered from this actually. The classic example is yeast converts sugar into ethanol, which is what makes the bubbles in beer. They're also important for making bread, and it's actually what makes bread rise. So the idea behind metabolic engineering is that we can use the same process to develop other useful chemicals, such as fuels, flavors, fragrances, or in this case, pharmaceuticals such as artemisinin. So to do this, genes to make a chemical of interest, like those found in the Artemisia plant, are moved into a simpler, easy to work with organism like baker's yeast. But doing this is not always easy, and there's actually a few problems that we need to overcome. So the first problem is identifying what genes carry out the chemical reactions of interest. We know the Artemisia plant produces artemisinin, but how? It turns out to be a really difficult problem because there are so many genes in a given organism, and there are many different techniques to find out which ones are relevant, but they're also often time-consuming and take many years to discover. The second problem is that organisms and proteins do not always have the exact traits we want them to have. So we have to look for variants of them that do, or create new variants that have the desirable traits. One example of this is that most people hate seeds in watermelons. Most sane people hate seeds in watermelons. So farmers found ways to grow seedless watermelons. And the reason that this is that enzymes in organisms are not naturally optimized for producing chemicals, sort of. Enzymes are optimized, but they're optimized for growing and sustaining life, as you might recall from such scientists as Darwin or Ian Malcolm from Jurassic Park. So the problem here is that enzymes are not optimized for having maximum activity in a cell. Nature's idea of optimal is not the same as a metabolic engineer's idea of optimal. This makes sense because oftentimes an enzyme might be producing an intermediate that the cell doesn't need large quantities of, or it might even be toxic to the cell. So to produce more of our desired chemical using metabolic engineering, we often need to do optimizations on these enzymes. So we can take this idea and use modern techniques to find useful mutations. There are two common ways of generating these useful mutations. First is to subject cells to something that mutates DNA, such as UV light, radiation, or chemicals such as benzene or sodium azide. You guys might have heard of these referred to as carcinogens because cancer often arises from mutations in the DNA. This is often represented by this little cartoon lightning bolt. A second, more targeted technique is the use of error-prone PCR. So Dan previously mentioned PCR as a method for amplifying DNA by replicating it many times within a test tube. The process goes something like this, where you have DNA, you prime it for replication with another small piece of DNA, and extend the primed DNA with an enzyme called DNA polymerase. So this is done cyclically, where the DNA polymerase extends and replicates the DNA, so you get many copies. So error-prone PCR has the same process, except we use a polymerase that has had its proofreading ability removed. So this leads to errors accumulating as the replication occurs, and this creates genetic diversity. So using these two techniques, we can generate many variants of enzymes and test them for our desired activity, such as enhanced production of a chemical. The third problem is that even after we combine the genes from one organism into another to produce a new chemical, such as artemisinin, we still have a lot of work to do. That's because, as you'll recall, in organisms, metabolism is extremely complicated. We need to balance the fluxes so we don't accumulate high levels of toxic intermediates. We also often have to make sure that there are not enzymes that interact with the new chemicals you're producing in the cell. So you might have to remove off-target interactions. And this is all very difficult and time-consuming and requires upregulating or downregulating the expression of different genes, including those that you added to the organism or that are inherent in the genome of the organism that you're using. And sometimes you have to remove genes completely. So it was facing these challenges that the Synthetic Artemisinin Project, or the ART project, was born in 2001 with the goal of using yeast to produce artemisinin. At the time, the idea of using a simple microorganism to produce something as complicated as artemisinin was an extraordinarily ambitious project. So who was crazy enough to try this? This person turned out to be Jay Kiesling, who was a professor of chemical engineering at UC Berkeley. He also wasn't alone with his ambition because he had to have another equally crazy person to support him in the project. So luckily for him, there was an extremely generous and wealthy person by the name of Bill Gates, who funded the project in 2004 through his philanthropic foundation. So Jay and his lab were able to successfully identify the key genes in the artemisinin pathway, port them to yeast, which I said before is used commonly to make things such as bread and beer, optimize the enzymes, and then balance the new pathways, the new metabolism, to increase the amount of chemical made. So this yeast strain developed is able to take glucose, a common sugar that you can grow. You can get glucose from different plants and use it to make artemisinin now. So the drug-producing strain has actually been licensed to Sanofi Aventis, and the synthetically derived artemisinin is now being used for treatments around the world today as we speak. So this work was a tremendous amount of effort, but it's pretty amazing that the synthetic artemisinin is being produced through fermentation, this totally novel process, to make a real-world impact with hundreds of millions of treatments currently happening. So quick recap, metabolic engineering is used to make useful chemicals, which is awesome, but it's difficult. You need to identify the useful genes, enzymes need to be optimized, and then metabolisms need to be balanced. Secondly, metabolic engineering was successfully used to change the way the world produces an important anti-malarial drug. And this is just a huge success story for synthetic biology.